The question reads, in brief state what happens when first is dry apricots are left for some time in pure water and later transferred to sugar solution. Now let us see the answer to this question. So apricots will swell up due to endoosmosis and then they will shrink due to exoosmosis. So when apricots are left in pure water, the water inside apricots is less and water outside the medium it is more. So osmosis will take place and water will move from higher concentration to lower concentration. So water will move from the medium into the apricots. So that is why apricots will swell up due to the water being filled in them. And this process through which the water is coming into the cell, this is known as endoosmosis. So when the dry apricots are transferred to the sugar solution, the solute concentration in sugar solution will be high and water will be less, and water inside apricots will be high, so water will move from higher concentration to lower concentration. That means water will move out of the cell, due to which the cell will shrink and exoosmosis will happen. That means that water will come out. Now the second part of the question reads, a red blood cell is kept in concentrated saline solution. Now let us see the answer. The RBC will lose water and shrink. So again exoosmosis will happen as saline solution consists more of the solute and less of the solvent, and RBC has more of the solvent. So exoosmosis will take place. That means water will move from higher concentration to lower concentration and RBC will lose all the water and it will shrink. The next part of the question says the plasma membrane of a cell breaks down. Now, if the plasma membrane of the cell will break down, the cell will die, because plasma membrane acts as a semi-permeable membrane and it allows the transfer of substances in the cell, and it also helps to keep a barrier between the environment and inside of the cell. So, if plasma membrane will break down, all these functions will not be performed and cell will die. The next part of the question reads, Rheo leaves are boiled in water first and then a drop of sugar syrup is put on it. Now let us see the answer to this question. So the cells of Rheo leaves are killed on boiling and hence there will be no plasmolysis. When the Rheo leaves are boiled, it will destroy the cell membrane that is present, and then when a drop of sugar syrup will be put on it, no osmosis will happen, because osmosis has one condition that it has to follow, that water molecules will move through a living membrane that is known as plasma membrane. So when we boil the leaves, the plasma membrane will be destroyed and there will be no plasmolysis or no osmosis will happen. Now next part of the question reads that Golgi apparatus is removed from the cell. So if Golgi apparatus is removed from the cell, all types of vesicle formation will stop. So in a cell Golgi apparatus, it has a function that all the material that is formed in the ER, for example the proteins are formed in the endoplasmic reticulum, so all these come to the Golgi apparatus and it forms vesicles like structure. So it has the function of packing the materials. So if Golgi apparatus is removed, all these functions will not be performed and vesicle formation will also stop. I hope you understood this question well. Thank you.